Hi, hello everyone, and welcome back. My name is Laura Perez-Brenner, your host of Learn and Burn. On this show, we ask scientists questions on life science technologies as they attempt to eat their way through a gauntlet of hot wings. Today's topic of conversation is measuring targeted protein degradation. And like usual, each of the wings will be doused in sauces made by the Promega culinary team, and they use locally sourced produce grown in the Garden of Promega Madison campus. And this time we have all new sauces. So let's see if we can still take the heat. I'm joined by Kristin Riching, a senior scientist and group leader who has been with Promega for almost 8 years. And Kristin has been leading the charge in developing approaches to monitor functional mechanisms of protein degradation and other key protein-protein interactions. All right, our first sauce is the buffalo bomb. This is prepared from cherry bomb peppers and guajillo peppers. It is low compared to the rest of the sauces, so since it only has 10,000 Scoville units of heat. All right, let's give it a try. This is good stuff.
This is nice.
Thank you, culinary team. All right, Kristin, what is targeted protein degradation and what are some of the challenges in design and optimization of degrader compounds, and how can our technologies address them?
Yeah, so targeted protein degradation, or TPD as we like to call it, is an emerging therapeutic modality for treatment of disease. And so historically, disease-causing proteins have been targeted through the use of small molecule inhibitors, which essentially function via an occupancy-based mechanism. So if you can, essentially bind up all the copies of a target protein in the cell, you can effectively inhibit and block the function of that target protein. But now we're actually seeing sort of the shift in targeting strategy, where rather than inhibiting the target protein, we now actually want to selectively degrade and remove all copies of that target protein from the cell. So this is what targeted protein degradation really centers on. And so it functions basically by, you can think of taking that exact same inhibitor, small molecule inhibitor compound, but then directly linking it to a second binding ligand, which directly recruits the cell's degradation machinery. And so that brings in this E3 ligase complex to generate a ternary complex between the target protein and the E3 ligase. And this then results in ubiquitination of the target protein, which serves as essentially a degradation signal, which is then recognized by the proteasome. So then the proteasome recruits that target protein, and that protein is degraded via the cell. So this is a very complex and dynamic process. It's very, very different from small molecule inhibitors. There are multiple steps in this pathway that have to occur in order for successful degradation to be achieved. And we call this event-driven pharmacology. And so the important thing in this context is the fact that this is a kinetic process. All of these steps have individual kinetic components to them, which can impact the overall efficiency of the entire process. So ultimately, what we want to do is be able to characterize how fast the degradation occurs, at what point the maximal degradation is reached, what is that maximal level of degradation, and how long that persists over time. Because if you were to look at a snapshot at a single time point or even multiple time points, they could look very, very different. And so it's important to have this kinetic perspective in understanding how efficient these degradation molecules are and characterizing their efficacy. So we've really built a platform of technologies that use our core luminescent technologies to characterize the degradation efficacy of these compounds. And this, we can do this by using our HiBit CRISPR technology. So HiBit is a binary complementation strategy based on our nanoluck luciferase technology. So it's a small 11 amino acid peptide that has high affinity for the large bit protein. And when those two subunits combine, they form a functional luciferase enzyme in the cell, a very bright luciferase. And because HiBit is so small, we can very efficiently insert it into the genome to track specific endogenous proteins and follow the kinetics of degradation. We can then couple that technology with nanobret technology, which we heard about in a previous episode related to target engagement. So this is a resonance energy transfer mechanism. But we can now look at the upstream mechanistic interactions along that degradation pathway. So we can specifically look at ternary complex formation, ubiquitination of the target protein, along with that endogenous target protein. And we can understand the kinetics of all of those processes. So we've really worked hard to build this platform of technologies to really enable both the kinetic aspects of degradation and understanding those aspects, as well as characterizing the mechanism of these compounds, so that we can further add to our understanding of these degradation molecules.
All right. Yeah. So the next sauce is called Sahara Smile, and it incorporates some Middle Eastern flavors with Peter and Thundercut peppers. And it brings some heat with 25,000 Scoville units. So we're increasing the hot scale. All right, let's give it a try.
I like that one.
Mm-hmm.
Yeah, I definitely, I feel the heat on that one a little bit. It was a big step up from the previous one, but not too bad.
Yeah,
that's good.
Kind of reminds me of harissa.
A little bit. I can see that. Yeah.
Yeah, it tastes good.
That a little bit.
Yeah, a little sip of water.
Yeah.
All right. So we have been talking about applications of luminescent technologies to explore proteosomal targeted protein degradation. Are these a method specific to that process, or can you apply them to other degradation mechanisms?
Yes. We definitely think that we can apply these same tools to studying other mechanisms of degradation as well. And given the success that we've already seen with these compounds that target proteins for degradation via the proteasome, we are actually starting to see application of basically the same idea, but using different mechanisms of degradation. So there are two main mechanisms of degradation that cells use, and one is through the proteasome, and the other is through lysosomes. And really, when people are trying to make these degrader molecules to eliminate proteins from the cell, it's really in an effort to try to pair a particular target protein with the most efficient strategy for removal. And so proteasome-mediated degradation is really great and works very well for soluble proteins, or proteins that are expressed in the cytosol in the nucleus. Say plasma membrane proteins are actually more efficiently degraded via internalization and fusion with a lysosome. Similarly, autophagy is a mechanism that targets larger protein aggregates and even cellular organelles for degradation. And so these growing autophagosomes envelop these structures and then ultimately fuse with the lysosome to facilitate degradation. So while the field is still very early on in showing successful examples of these different targeting strategies and targeting specifically to the lysosome, there have been several successful examples. And we definitely think that these same luminescence tools can be applied in this case, because definitely kinetics will continue to be an important driver of these mechanisms, and it will be important to characterize the kinetics in order to understand the efficacy of that degradation. So we definitely think that these same tools will be important. But some of the differences could depend on the precise recruitment strategy. And so thinking about and building the precise mechanistic interaction assay and how we think about that recruitment strategy, there will be some likely nuances there in terms of that development. But this is exactly the same place that we started in when we first started in this area of protein degradation. So I definitely think some of these challenges can be addressed, and we can definitely apply these same tools that we have.
All right, ready for some heat?
All right. I don't know. I'm still feeling that last one a bit, actually. I was surprised.
Me too. I had a cough attack also. Well, that's not a good sign, especially because we have like 3 others. So the third in line, we have the Avanada Rush at 50,000 Scoville units. It's funky, fruity, and a little bit fiery. So let's see how fire it is.
See, it's got some peach in it.
Yeah, and it has a super bright color. Love
It
it.
does. Well, it's a
Maybe
little
it's like a
chunky.
warning. Yeah, it's like a warning sign. Maybe like, be careful. I'm gonna give it a try.
Definitely hotter.
It is.
I don't think I get the peach.
It's like, yeah. I feel more of the fruity flavor on the background.
Oh, now I'm getting
Yeah.
it a little bit.
Mm-hmm.
Hmm.
It's very good.
That is good. Yeah.
So the other one it left me coughing pretty.
I felt it initially, but now it's gone. Yeah.
Yeah. Also, I put a
Hmm.
lot less on this one because I'm like, well, if I cough that much for the other one, then I should like balance it out. All right. Shifting gears, it is more important than ever to grow our efforts to build diversity, equity, and inclusion in the science world. What do you think we should keep in mind to encourage engagement and also eliminate barriers to entry in the chemical biology field and beyond?
Yeah, this is a really important question and one that I've personally given a lot of thought to. And for our viewers who are joining us, I am actually filming with my African gray parrot, so you may actually hear some irreverent interjections from him. But that is my parrot. But yeah, getting back to the focus here. In order to try to eliminate some of these barriers, I think it's important to recognize that not only why these barriers exist in the first place, but also when they start to show up. And in many cases, we see these barriers are present in childhood and into young adulthood, and they only get bigger as time passes. And so, a child who is interested in science or maybe looks up to a scientist, but never has that opportunity to really experience what it's like to maybe be a scientist or experience that fun and creativity of designing an experiment and learning something potentially new about the world, that can ultimately lead them to losing interest in science in general. And the other thing that I think can sometimes be difficult for people to recognize is that it often feels like the responsibility is on the person experiencing the barrier to try to come up with strategies to eliminate that barrier. And it's so easy to say if you're not the one experiencing it that you're going to just disengage from that conversation entirely. But I think more and more people are starting to recognize that the responsibility isn't on the people experiencing the barrier, it's on the rest of us to really work together to try to build equity and in both science as well as in other areas of life. And I think that there is some good work being done here. Our group last fall actually had a chance to connect with and mentor a high school student and bring that student into the lab and give hands-on laboratory experience working on a project that actually had some potential clinical significance. And so that's an experience that most students would not have. Certainly would only be possible if, say, a student had a parent who was also a scientist, or if they happened to attend a school with significant resources available to them. And so this is something that I'm really hopeful that we'll be able to do more of and expand and give these other opportunities for. But then outside of my job, one of my main passions is mountain biking. So I'm actually a coach on the local youth mountain bike team here in Madison. I've been a coach for the last 4 years. And mountain biking is, a sport with incredible barriers to entry. And this was also apparent on the team, certainly with respect to lack of diversity on the team. And we recognized that this was due to problems of access. So, access to bikes, access to trails, access to a safe environment in which to learn. And so we got together and we put together an event to try to give kids the chance to try out mountain biking if they didn't have a mountain bike or if they'd never mountain biked before. This would be a place where they could come and just try it out and have fun. And we partnered with local groups to provide bicycles to the kids, and this ended up turning into an event that we held annually, and also turned into a program that gave kids the chance to connect with the team and provide them with opportunities to then access and procure bikes that they could use for the season to participate on the team. And so there really is some good work that people are doing, and I'm hopeful that we'll see more work, more examples like this going forward, and particularly with respect to providing opportunities for youth.
That is excellent. And I love the two angles, like, while at work, but also outside of work. It's super important to keep that in mind and do something about it, right? Recognize like our current gaps and then breaking barriers and making hopefully like a future much different than what it is. So I applaud you for that.
Thank you.
The name of our next sauce is misleading. It's called Cool Runnings, and it should be pretty hot at 125,000 Scoville units. Are you ready to try it?
I'm ready.
All right.
Yeah.
Hopefully I would not put too much. Oops. Oh, I think I put too much. All right.
I'm not getting any spice.
This is really good.
I really like it, actually.
Mm-hmm.
I love the flavor.
It tastes like kind of like allspice jerk chicken. It's really good.
Definitely getting the fruit. Oh, now it's kicking in. Oh, wait a second. Okay.
A delayed response. Those are the worst.
Yeah. That was definitely a slow
Yeah,
build.
it's not like all hot. Yeah, it's not all hot. It actually tastes really, really yummy and kind of Caribbean flavors. In addition to the ASIC technologies, what are some of the offerings in Promega's Taylor R&D Solutions group that can enable discovery and screening of compounds for degradation?
Yeah, we have a really fantastic team of scientists as part of our tailored R&D Solutions group that is really dedicated to providing a lot of custom services in the area of targeted protein degradation. So this encompasses cell line generation, so those high-bit CRISPR edited cell lines that I mentioned earlier. So we have a group that is dedicated to building those custom cell lines and enabling monitoring of target protein levels. But then we can also do compound profiling services. So can we try to identify the next generation degrader compounds and new candidate molecules using our technologies? So we can screen for degradation in either endpoint or kinetic fashion. So as I mentioned before, all of the advantages that kinetics provide, we're able to run those profiling experiments, and we also have all of the tools to enable the quantitative understanding and quantitating all of those degradation parameters for rank ordering the responses and those compounds to evaluate the efficacy. We can then also take those hits and validate the mechanism of action. So as I mentioned, our nanobret technologies, we can also profile compounds using those technologies as well. So we can screen for ternary complex formation as well as ubiquitination, and also just validate that the compounds are on mechanism and functioning through the intended pathway. And then lastly, we also provide services for de novo assay development. So for new targets that we maybe have never worked with, or potentially new E3 ligase molecule or E3 ligases that these molecules might recruit. And in addition to these new degradation modalities that I mentioned, there's also some de novo assay development that we do in those areas as well. So this would be, again, combining maybe novel targets with novel recruitment strategies. So we provide a wide range of capabilities and service offerings in this area.
Excellent. Looks like we have a lot of different offerings. So if anybody is interested, contact us. All right, we made it to our fifth and final sauce, and it has a little bit of a scary name. It's called Fatali Fallout. And I hope that you're ready to try the Fatali pepper, since they're the sixth hottest pepper in the world. So buckle up for 350,000 Scoville units.
All right. I don't know. Just in case, I've got my ice bucket here with some ice cream. So I don't know. Maybe I'll take a preemptive bite here of ice cream. Also ready for the ice bucket challenge at the end of this.
Go big or go home.
Exactly. Okay. I feel like I've gotten off easy with these so far, so I think I'm just going to go for it with this one. I don't know.
Mm-hmm. Mm-hmm.
Waiting for it to build. Waiting for the moment that it takes me out.
I put more chicken than hot sauce. I'm gonna go wild now.
I've never put in context. Nugget here that is
Okay.
minimizing the effect.
Okay. Yep.
Yep, I'm getting a bit more.
You'll feel the heat for sure. Well, I guess I will survive the challenge. Maybe not.
I might actually go for the ice cream now. Yeah, I'm just
Maybe
gonna
I should
do
go
a quick
for the
bite
red.
here. This was my excuse all along. Oh yeah, that took it right
I
away.
wish I would be like as smart as you bringing the ice cream, but I didn't. I thought it was gonna be tough, but not really. But that was hot.
I can still feel it a little bit in the back of my throat, but not too bad.
Now that we made it through, you have the audience undivided attention. So please give a shout out to our latest news or what's on the horizon for your work on targeted protein degradation.
Yeah, we have a lot of new exciting things going on in targeted protein degradation right now, specifically at Promega. And in I think the previous episode, we heard a lot about LUMET technology, and we're really excited about applying LUMET technology now to protein degradation. So, and this would be specifically to look for compounds which can form a ternary complex, ternary complex, but in a homogeneous biochemical format. So if we have compounds that are, say, impermeable and can't get into the cell, how can we at least study whether or not they can form ternary complex, and then we could optimize for permeability, at a later point. But so this LUMET technology, we're working on making ternary complex assays with to specifically look at exactly this, whether compounds can just form ternary complex in a biochemical format. Some other things we're working on are expanding applications of haloprotac. So haloprotac is a tool molecule that we can use to study degradation of any target that is fused to halotag. So haloprotac recruits halotag fusion proteins to VHL E3 ligase and then targets them for degradation. And this is a compound that is also useful for not only evaluating the ability of a particular target protein for degradation if there are no small molecules that bind to that target protein that could be used to make a specific degrader with, we can enable study of those targets just with haloprotac, but we could also look at the resultant phenotype. After we degrade that protein, we can look at the phenotypic consequence that has on the cell. So we're looking at expanding some of these applications of haloprotac. And then lastly, we talked about these other degradation targeting strategies that we're starting to see some successes with. And we're also very active in this area as well, both from the standpoint of trying to provide ways to enable development of those different degradation strategies, but also to enable characterization of the mechanism of action of those strategies. So there's a lot of work that we have going on, and we're really, really excited, and yeah, stay tuned for more developments.
That's awesome. Super excited about that. All right, so that's the story for today. So Kristin, please take some time to recover, and I would also take some time as well. And thank you so much for sitting down to talk science and eat heat, so stay sizzling.
Yeah, he was, I think, anxious to try some veggie nuggets, so that's hence the screaming and the crow noises.